Multiphoton microscopy of fixed tissue is normally limited to shallow penetration depths of just a few hundred microns. We recently demonstrated the first use of multiphoton microscopy as deep as several millimeters into fixed mouse organs using optical clearing with a solution of benzyl alcohol and benzyl benzoate. Our initial demonstration of the technique imaged intrinsic tissue fluorescence. However, there is increasing interest in using optical clearing methods with tissue-expressing fluorescent proteins such as YFP. Here, we present a protocol for multiphoton microscopy of cleared brain tissue with the same benzyl alcohol solution that preserves the fluorescence of yellow fluorescent protein expressed by the Thi-1 promoter in mouse brain.
High-resolution, whole mouse organ imaging using multiphoton microscopy is made possible by optically clearing the organ before imaging. Without clearing, multiphoton imaging of the brain is limited to 300 microns beneath the tissue surface due to high light scattering effects created by the differences in refractive indices of water and the proteins that make up the brain tissue. To overcome this limitation, the organ is dehydrated and the water is replaced with a fluid that has a similar refractive index as the proteins that make up the tissue. This process of optical clearing helps to greatly reduce light scattering to allow for better imaging farther beneath the tissue surface. Images from Parra et al. Demonstrate how this technique can be applied to view the histology of different mouse organs using intrinsic fluorescence and second harmonic generation. This first image is of the mouse testicle taken 1.4 millimeters beneath the tissue surface. The high-resolution property of multiphoton microscopy allows us to zoom in and get a clear look at the individual sperm cells forming in the seminiferous tubules as seen on the far right. Here we show an image of the mouse lung, which was also taken 1.4 millimeters beneath the tissue surface. This image is a demonstration